Okay, hello everyone. So today one new topic. We continue with the metabolism of carbohydrates. So we have completed the story on crabs and degraded glucose to carbon dioxide and water, but now we have to focus on one other catabolic pathway, which is called Hexose monophosphate pathway or pentose phosphate cycle or phosphogluconate pathway, so numerous names for the same metabolic pathway. So let's provide an overview of this pathway, and let's go briefly through some facts that we're gonna focus on in detail in the upcoming minutes. So this hexose monophosphate pathway actually occurs in all cells, and within the cell it occurs in total in cytosol. So this pathway actually includes two major parts of the pathway, or two major domains of the pathway. So first it includes two irreversible oxidative reactions, and after those two are followed by a series of reversible sugar phosphate interconversions. What can we expect out of this hexose monophosphate pathway? First, let's explain one very important fact. This is considered to be catabolic pathway because precursor glucose 6-phosphate is converted to some other monosaccharides containing less than 6 carbon atoms. So it's considered to be a catabolic pathway. But the very important thing is that during this catabolic pathway, no ATP is neither directly consumed nor produced. So it is catabolic, but it's not energy yielding pathway. So during this pathway, one carbon atom from glucose molecule, and that is C1 carbon atom, is removed and it's released in the form of carbon dioxide. The second aim of this pathway is the formation of NADPH coenzyme. So two molecules of NADPH are derived from each glucose 6-phosphate molecule actually entering this hexose monophosphate pathway. This is important because actually this pathway provides a major portion of the body's NADPH. Which role is, and we're going to see that in a couple of minutes, is to serve to our cells as a biochemical reductant for biosynthetic pathway or in the processes of reductive detoxifications, which are going to be explained later. Also, one of the products of this pathway is ribose 5-phosphate, which is necessary precursor for nucleotide metabolism. So multiple purposes of this metabolic pathway, and let's start from the beginning. So I have already mentioned that this metabolic pathway starts with two irreversible oxidative reactions. Actually, two parts, but three reactions, which are defined as oxidation reaction and decarboxylation reactions. And the final products of these three reactions are the formation of ribulose 5-phosphate, carbon dioxide, and two molecules of NADPH. Those three types of molecules for each G6P oxidized molecule. So this oxidative part of the pathway is particularly important in liver, lactating mammary glands, and adipose tissue. Why? Because in these tissues, enhanced intensive fatty acid biosynthesis occur, and you will see later in some other lectures, those are NADPH requiring metabolic pathways. Also, this pathway is so important in testicle, ovaries, placenta, adrenal cortex. Why? Because in those tissues, steroid hormones biosynthesis takes place, and for that biosynthesis, again, we need NADPH coenzyme, which is provided by this pathway. And further on, and it's going to be emphasized later on as well. It's particularly important in erythrocytes because they require NADPH to keep reduced glutathione levels at certain physiological level and as a front line in the defense of oxidative stress. So now that we know that these pathways are so important, let's explain which reaction occurs during the hexose monophosphate pathway. And here is the figure which I really like a lot, and I think it's very illustrative and explains the whole metabolic pathway in a simple way, an understandable way. So I would like to use and take a moment with this figure to actually make an overview of the overall metabolic pathway. So let's start. So this is the major precursor, glucose 6-phosphate. So this glucose 6-phosphate is going to be oxidized by the enzyme glucose 6-phosphate dehydrogenase in the reaction number 1 of the oxidative part of the metabolic pathway. And during that process, one NADPH molecule is synthesized, as you can see, and what you cannot see here is that 6-phosphogluconolactone is formed. But this is not a very stable molecule, so it's readily hydrolyzed by another enzyme, 6-phosphogluconolactone hydrolase to phosphogluconate. So this is the part 1 of oxidative domain of the metabolic pathway. In the reaction number 3, or in the second part of this oxidative part of the pathway, decarboxylation occurs and another oxidation yielding the formation of the second NADPH coenzyme, which can be used for reductive anabolic pathways or for antioxidant protection. This reaction is catalyzed by 6-phosphogluconate dehydrogenase, and if you can see, one five-membered ketose is formed, one ketopentose is formed, which is ribulose 5-phosphate, which can further be converted to numerous monosaccharides containing 3 to 7 carbon atoms, and you can see it can be converted to ribose 5-phosphate, which can be used for nucleic acid biosynthesis, or it can be converted to cellulose 5-phosphate, which is going to be one very interesting intermediate of this pathway. Then we have two other enzymes, transcatalase and transaldolase, which are responsible for the transfer of 2 carbon fragments or 3 carbon fragments from one monosaccharide to another. And this is one of the examples. Cellulose from cellulose we can transfer 2 carbon fragment to ribose 5-phosphate to get sadoheptulose 7-phosphate and glyceraldehyde 3-phosphate, which is an intermediate of glycolytic pathway. Then transaldolase can transfer 3 carbon fragment from sadoheptulose to, for example, glyceraldehyde 3-phosphate to give erythrose 4-phosphate and fructose 6-phosphate, again one of the intermediates of glycolytic pathway. And so on. There are numerous possibilities for interconversions of those sugar from one into another to provide either those major final products or intermediates of glycolytic pathway, depending on the actual and current cell needs. But we're gonna go one step after another, explaining this pathway in details. So the first event that takes place in hexose monophosphate pathway is dehydrogenation of glucose 6-phosphate. So here is the reaction extracted from the previous figure. So the enzyme glucose 6-phosphate dehydrogenase is the one which catalyzed this oxidation. So glucose 6-phosphate is oxidized to 6-phosphogluconolactone, and during this oxidation one NADPH reduced is produced, so the first out of 2. This enzyme, remember, is the main regulatory rate-limiting enzyme of the pathway. And NADPH is a potent competitive inhibitor of this very enzyme. So we have an example of feedback inhibition. Insulin upregulates expression of the gene for this enzyme, glucose 6-phosphate dehydrogenase. The second reaction of this first oxidative part, is the hydrolysis of this phosphogluconolactone and its conversion to 6-phosphogluconate, and the enzyme is 6-phosphogluconolactone hydrolase, which is not a rate-limiting enzyme. The second part of oxidative domain of the pathway is formation of ribulose 5-phosphate, if you remember. And the third reaction of this part of the pathway is irreversible reaction, of course, like the previous one, but this time this is not a regulatory reaction. Actually, this is an oxidative decarboxylation of 6-phosphogluconate, which means that one carbon dioxide is removed from the carbon number 1 of glucose, and we get ribulose 5-phosphate, which is a pentose phosphate molecule, a ketose molecule, and the second NADPH, the last NADPH, is formed. The mechanism of this reaction is very similar to isocitrate dehydrogenase reaction taking place in Krebs cycle, if you remember. After these pathways, the sequences of reversible non-oxidative reactions occur. So this non-oxidative part of the metabolic pathway occur in all cell types synthesizing nucleotides or nucleic acids. So this part of the metabolic pathway actually catalyzes the interconversion of different sugars, different monosaccharides containing 3 to 7 carbon atoms. So if you remember from the previous figure, ribulose 5-phosphate is either converted to ribose 5-phosphate, which is a precursor and starting point for nucleotide synthesis, or ribulose 5-phosphate can be converted to glycolytic intermediates, fructose 6-phosphate and glyceraldehyde 3-phosphate. So for that purpose, we have to employ two enzymes, transcatalase and transaldolase, because let's be realistic, most of the cells, in many cells, there is a greater need for dose reducing equivalents NADPH rather than ribose 5-phosphate for nucleotide biosynthesis. So transcatalase is an enzyme which I've already mentioned, responsible for transfer of 2 carbon units of keto sugars in thymine pyrophosphate dependent reaction. So we need a TPP as a coenzyme, and the role of this coenzyme is very similar to that one which we've explained in oxidative decarboxylation of pyruvate, and the one which we explained in the alpha-ketoglutarate dehydrogenase reaction. Transaldolase is another enzyme of this non-reducing, non-oxidative reactions, and it's responsible for the transfer of 3 carbon fragments and converts ribulose 5-phosphate to glyceraldehyde 3-phosphate and fructose 6-phosphate intermediates of glycolytic pathway. At this time, transaldolase activity is very similar to the activity of aldolase in glycolytic pathway, and the names are also similar. So in case that cell needs more ribose than NADPH, for example, undergoing intensive mitosis, then non-oxidative reactions are favored to synthesize ribose 5-phosphate rather than NADPH coenzyme. Which of these directions are going to be favored depends on the current needs of a cell. So transcatalase activity determination in erythrocytes is one very interesting laboratory tool for measuring thymine nutritional status, especially in order to be able to diagnose the presence of thymine deficiency. So in order to do that, the activity of transcatalase is determined in the presence and in the absence of added TPP. So in persons with an adequate intake of thiamine, no increase of activity of this enzyme will be observed because this enzyme already contains sufficient amounts of TPP. But in thiamine deficient patients, at the beginning, the enzyme will exhibit rather low activity, but after and upon addition of TPP, it's going to be greatly stimulated, indicating that there is a thiamine deficiency and that there are some active sites and parts of the enzymes which are not saturated by sufficient amounts of TPP. Another example of the usage of these intermediates of hexose monophosphate pathway is a very interesting molecule of cellulose 5-phosphate, which is a molecule which does not do only interconversions of sugars, but it also regulates gene transcription. Why is that so interesting? Because, for example, in the states of excess intake of carbohydrates in diet, we all know that all excess amounts of sugar are going to be converted into lipids, predominantly 3-acylglycerols. So when there is a large or huge influx of glucose into the liver, it's going to, of course, logically increase the levels of X5P in cells. Increased X5P is going to activate protein phosphatase 2A, and this enzyme is going to dephosphorylate one transcription factor, CHREBP, or carbohydrate response element binding protein, which is actually going to bind to specific promoters which are going to start transcription and upcoming translation for enzymes. And take a look at this. Pyruvate kinase, which is the rate limiting enzyme of glycolytic pathway, or malic enzyme, citrate lyase, acetyl coenzyme A carboxylase, fatty acid synthase, all of those enzymes for fatty acid biosynthesis. So what is going to be the final result of this effect of X5P? It's going to be an increased biosynthesis of fatty acid. Thus, as a consequence, increased biosynthesis of 3-acylglycerol and their subsequent incorporation in VLDL particles in liver and their export into the bloodstream, where an increased level of VLDL can be measured or determined. But now that we know those details about the pathway itself, let's focus on NADPH a little bit. Let's focus on its properties and functions and roles in our body. So NADPH is involved in reductive reactions rather than NADH. And to contribute to this fact and information, we can say that the ratio of NADPH to NADP plus is much greater than reduced NAD to NAD plus. I'm going to explain this. So you know that we get NADH from oxidative processes, and when we get NADH, it gives reducing equivalents to ETC, and it's rapidly oxidized back to NAD+. This is catalyzed by NADH dehydrogenase in ETC. So you get a formation of NADH in catabolic pathways, and they readily give away their reducing equivalents. So the level of NAD reduced in cells is rather low. On the other side, NADPH reduced can be produced not only by hexose monophosphate pathway, but in liver and some other tissues. There are some other metabolic pathway which are capable of formation of NADPH, of course, but the red blood cells, erythrocytes, are exclusive for hexose monophosphate pathway. Also, the third option for NADPH to be synthesized is that in mitochondria, There are some enzymes which are transhydrogenase, and they're energy requiring enzymes which are very close to the complexes of ATC system, so they can transfer reducing equivalents from NADH to NADP plus, thus forming reduced NADPH. So you have three possibilities to get NADPH in a cell, and you have only one possibility to get NADH, and it's readily oxidized to NAD plus again. On the other side, NADPH, if we remember bioenergetics, cannot be oxidized directly by ATC. So, as a conclusion, in a cell, the ratio between reduced NADPH and oxidized NADP plus is greater than 1. Which is important because this ratio actually contributes to the energy which is needed for biosynthetic pathways, and at the same time it provides a constant source of reducing power for detox reactions. Which are the functions and roles of NADPH? So we have mentioned, and that is what we all know, that coenzyme NADP plus differs from NAD plus only by the presence of this phosphate group at one of the ribose units, at the position number 2. So this obviously small difference in the structure of this molecule actually directs the role of these molecules in a cell. It actually favors the usage of NADPH in reductive biosynthetic processes, like biosynthesis of fatty acid, which I've already mentioned, or the biosynthesis of cholesterol, and so on and so on. And one more, NAD and NADH favors oxidative role of NADP plus, while NADPH favors reductive role of NADPH. So to conclude, something that that you probably know, electrons of NADPH are destined primarily for use in reductive biosynthesis rather than for the transfer of oxygen to oxygen in ETC, as it is the case of NADH. So this is one of the dominant role in reductive biosynthesis. But this is not the only one. Another one is also important, and that is the reduction of hydrogen peroxide. And this is something that most of the students struggle, and it is about reactive oxygen species, free radicals, damage of cell membranes, oxidative stress phenomenon, antioxidants. And now I'm going to take just a few seconds to go into this topic, but for the purpose of explaining the role of NADPH. So what do we know about hydrogen peroxide? We know that it belongs to the family of reactive oxygen species, ROS, which are formed by a partial reduction of molecular oxygen. And if you take a look at the figure, you can see that subsequent conversion of molecular oxygen to superoxide anion radical, and then hydrogen peroxide, which is further reduced to very potent and reactive hydroxyl radical to be finally reduced to the molecule of water. So these reactive oxygen species, be aware of this, are normally and continuously synthesized during normal aerobic metabolism. So they are formed as byproducts, but this is normal and this is the physiological conditions. But these quantities of ROS can be very efficiently neutralized by cell's defense system. Apart from the normal metabolism, ROS can be synthesized in the reactions with medicines or some xenobiotics, environmental toxins, or when the level of antioxidants are diminished for any reason, creating the condition of oxidative stress, which actually represents the disbalance between the overproduction of ROSs and, on the other side, diminished antioxidant defense system. So, ROS, those reactive oxygen species, can cause severe chemical damage to DNA, to proteins, to unsaturated lipids, which at the end may lead to the cell death. So it is believed that ROS have been implicated in a number of pathological processes, including reperfusion injury, carcinoma, inflammatory diseases, aging, and so many others, neurodegenerative diseases, cardiovascular diseases, and so on and so on. So we have a problem with ROS, and hydrogen peroxide is one of them. But luckily for us, our cells have several protective mechanisms which serve to minimize the toxic potential of these agents. And one of these potential molecules is reducing glutathione or reduced glutathione, GSH, or gamma-glutamyl cysteanylglycine. If you remember, we mentioned this molecule as one of the biologically important peptide molecule. It's found in most cells, especially high concentration of GSH are found in liver, because most of the detox processes occur in liver. So this molecule is capable to chemically detoxify hydrogen peroxide. This reaction is catalyzed by, I'm going to get back to this figure, is catalyzed by glutathione peroxidase, so which uses reduced glutathione to neutralize hydrogen peroxide and reduce it to the molecule of water. And during this process, reduced glutathione is oxidized to oxidized disulfide form of glutathione, which does not have protective properties anymore. So it is important for the cell to regenerate reduced glutathione, so to convert oxidized to reduced form again. And this is achieved by one other enzyme, which is called glutathione reductase, and this enzyme requires NADPH as a source of reducing equivalents. So this is the reason how NADP is indirectly involved in the reductions and the transfer of electrons for reduction of hydrogen peroxide and in those antioxidative defense mechanisms. Of course, it's not the only pathway of getting rid of those ROSs. Some other enzymes like superoxide dismutase or catalase are also enzymes catalyzing the conversion of other ROS like superoxide anion radical or hydrogen peroxide, again for catalase, to harmless products. So they all together as a group, they represent a cell's defense system against toxic effects of ROS, and our NADPH from hexose monophosphate pathway plays an important role in this system. I'm going to get back to the fact that erythrocytes are totally dependent on pentose phosphate pathway for their supply of NADPH, because unlike other cell types, erythrocytes do not have any of those previously mentioned alternative sources of this essential coenzyme. So either hexose monophosphate pathway is functional, or erythrocytes are gonna lack NADPH coenzyme, which is of essential importance for antioxidant defense system and for protection of erythrocytes from the overaccumulation of ROS, because erythrocytes are so exposed to oxidative stress and the overproduction of ROS because of the significant amount of oxygen which is found in erythrocytes attached to hemoglobin. So this is the figure which actually explains how any defects in hexose monophosphate pathway, and especially glucose 6-phosphate dehydrogenase deficiency, which is a regulatory enzyme of this pathway, may do a lot of harm to erythrocytes, causing its damage and complete depletion and hemolysis. So in order to maintain the normal function of cell membrane in erythrocytes, you need NADH and you need ATP, which erythrocytes can get from anaerobic glucose. And this is represented by the pathway number 1. So we have the glucose converted to glucose 6-phosphate, and further glucose 6-phosphate can enter glycolytic pathway, ending in the production of ATP and NADH. Of course, and lactate, because we mentioned anaerobic glycolysis is found in erythrocytes. On the other side, phosphorylated glucose can enter, as we saw previously, hexose monophosphate pathway to provide significant amounts of NADPH coenzyme by the action of glucose 6-phosphate dehydrogenase as the main regulatory enzyme. Previously, we mentioned that NADPH is of crucial importance for the regeneration of reduced glutathione by the action of glutathione peroxidase and glutathione reductase. And further on, we mentioned that reduced glutathione is used for neutralization of the overproduction of hydrogen peroxide, which can be overproduced either by spontaneous processes of oxygen in erythrocyte itself, or in the presence of some infections or the usage of certain drugs and so on. So if there is a deficiency in glucose 6-phosphate, this pathway is compromised, meaning that there is not going to be sufficient amount of glutathione in its reduced form to neutralize the overproduction of hydrogen peroxide and other reactive oxygen species. Thus, they can be converted to very harmful hydroxyl radical, which can chemically lead to the hemolysis. On the other side, oxyhemoglobin can be converted to methemoglobin, and due to the overproduction of oxidized form of glutathione, Heinz bodies are formed, which actually tend to enter the capillaries to pass through membranes, so they actually represent some kind of mechanical attack to the cell membrane. So combination of chemical and mechanical attack to membrane may lead in the end to severe hemolysis in those patients with glucose 6-phosphate dehydrogenase deficiency. Of course, we have to say that this can be compensated to a certain extent by antioxidant vitamins E and C, or by catalase, which is an antioxidant enzyme. So most of these patients are actually asymptomatic unless they are exposed to some oxidative challenge in which ROS is going to be overproduced. But apart from these, there are some other roles of NADPH in our body, so let's name them, or let's list them. So NADPH is required for cytochrome P450 monooxygenase system, and this is actually NADPH is a source of reducing equivalent for hydroxylation of numerous compounds such as aromatic compounds, steroids, alcohol, and a number of drugs and medicine by cytochrome P450, but detailed story will be provided in a chapter on liver biochemistry. Apart from this, NADPH is involved in phagocytosis process, and an NADPH oxidase actually uses NADPH to form superoxide anion radical from molecular oxygen. Why is that so important in phagocytic cells? Because superoxide anion radical then regenerates or generates hydrogen peroxide, which you saw from the previous figure. And hydrogen peroxide is the one very popular nowadays in the scope whether it can kill or not coronaviruses. But in our system, in phagocytotic cells, actually hydrogen peroxide is responsible for killing the microorganisms which are taken up by our phagocytotic cell and white blood cells as well. And the last role of NADPH worth mentioning here is its involvement in the biosynthesis of nitric oxide, which is very important for vasodilation. But the overproduction of NO can lead to the formation of some nitrogen reactive species and the formation of nitrosative stress, but that is going to be reserved for the story with one other lecture. So anyway, my dear students, my dear colleagues, that was all that I wanted to tell you about the hexose monophosphate pathway and the importance of NADPH for the overall metabolism. I hope the story was interesting and useful. I hope it was understandable. So until some other time and until some other topic, bye bye.